Hmm. Can somebody tell me if I talked about grading in the lecture? I need to know if I need to talk about it in the lab.
You haven't talked about grading yet.
Okay, I'll do that now. Thank you. Let's see, I don't need that. Let's see what that, excuse me. All right, let's go to the grade book and I'll talk a little bit about grading. Hopefully I'm not sharing my screen because I'm going to be putting the grade book here. It just helps me talk about it. I got a lot graded this weekend, a lot to still grade, but I got some of the extra credit study group sessions graded all the way through 5. I got lecture worksheet 4B graded. I've started grading lab. Is it 5? Started grading lab 5, but I've only got about a third of the students graded, and I'll try and work on that tonight and finish lab 5 for tonight. I doubt I'm going to get to lab 6. So that assignment is probably going to be postponed for another week. Not the assignment, but the grading. And then the regrading will be postponed to second week. But I'm talking about lab 6. That's why I changed the schedule, and I'll talk about that in a minute. I have practice quiz 00A graded and quiz 00B graded. You have to see the quiz 00A, you have to click on the icon of the file and it should show you your extra credit grade that you got for that. Assuming that you did that quiz. For some reason, this row in Canvas has a problem, and it's been a problem in the past, and it's not showing you the grade. I think it's not showing you the grade in the current score, but it is showing you the grade in the total score. Anyways, you can see the grade by just clicking on it. That was extra credit up to 1 point. Quiz 00B. I think I got practice quiz A 00A graded earlier. And I didn't realize that it hadn't been graded because it's showing that it hasn't been graded yet. It's showing the blank documents for mine. But if you click on it, you can see the grade. And then quiz 00B grade practice quiz 00B, I did get that graded. It's 0.1 correct, 0.1 point for each correct answer, up to a total of 1 extra credit point. Getting back to lab 6, I'm not going to get it graded, and you were going to have a quiz that would cover lab 6 tomorrow. And I decided initially I'd put up an answer because I wasn't going to get it graded in time. But then I realized if I put up the answer, there will be no point to having a second grading of that quiz. So I decided instead to postpone the quiz by 1 week. And that'll give me another week to get lab 6 graded. And that required me to also postpone quiz 4 by 1 week. Anyways, I hope you're satisfied with that. I just didn't want to quiz you over something I hadn't graded yet. Any question on any of that?
Yeah, on mine gradebook, it still says that quiz 00A is not graded.
No, click on it and it'll show you. Click
So
on
I
the
click
file.
on it, that's the actual grade.
Yeah, it'll show you the actual grade if you click on it. You have to click on the spot though. It is
Okay,
graded. Okay.
all right.
All
Thank
right.
you.
And like I said, there's a problem in that. I've seen it before. It's a bug in Canvas, and that that column, it's not behaving correctly, but it is part of your total points. So it will count. And at the end of the term, you will get the credit from it. But at least in your current points now, I don't think it's counting. And I don't know why, because I graded it. I actually graded that, I think, last weekend. All right, that's enough for the grade book. Let me get out of here. And I've talked about the postponing the quiz. Let me go back here. I have redone the schedule, which I'll show you in a minute. Although I haven't put up the new syllabus, I need to do that. And I have updated the website, so it is showing you the quizzes in the correct weeks, meaning that the quizzes are being postponed by 1 week. Any question about any of that?
I had a question about the unknowns project. I did message you on Canvas, but I'm still confused about where we're getting the email from you about the first three.
I haven't sent it out yet for the Tuesday, Thursday people. I will after the lab sometime tonight. I'm not going to
Oh,
promise it's going to be early though, because with the Monday, Wednesday people, I did finally send out the email, but it was after midnight. Okay, a little
oh,
bit after
okay.
midnight. So sometime tonight, I will send you your unknown
That's
number and your first three test results. My goal is to figure out how to send one email to the 3 o'clock lab people and a separate email to the 7 o'clock lab people. The trouble is I merged the labs and I'm not sure how to do that. If I can figure it out, I'll do it that way. But if I can't figure out how to do it, you're going to get an email that'll have two files, one for the 3 p.m. people and one for the 7 p.m. people. And hopefully I can figure it out. But if I can't, you're going to have to deal with a slightly confusing email. Just look at all the 7 p.m. Information for you guys.
okay, sounds good. And if we, I just didn't want to get behind. And then if we decide that, so I go to the labs interchangeably depending on what works best with my schedule, will that be an issue?
That
If I'm
shouldn't be an
signed up,
issue. You will be signed up for what you registered for.
got it. Okay,
Okay.
thank you.
Let me see if I can put the syllabus in now. Here it is. If I can find the syllabus, I will upload it right now. There it is. And you guys are going to be 2-3. So hopefully I just uploaded the syllabus now. I'm not actually going to check, but I've got it for Tuesday, Thursday. Now need to do it for Monday, Wednesday, but I'll do that later. All right, let me get out of here and let's go take a look at the syllabus. And I gotta share my screen. You should be seeing my screen now. So this is the new schedule, and we don't have a quiz tomorrow. Quiz 3, it's going to be postponed for 1 week because it's covering lab 6, and I don't have that graded yet. What we're doing today is lab 7, throat and urine cultures, and hopefully we will get the unknown, although I'm not going to promise that it'll be early, but sometime tonight I'll send out the unknown number as well as your results for the first 3 tests. Any question about what we're doing? I think I'll go ahead and leave that up. Let's move on to the throat and urine cultures. That looks like I might have the wrong one. Nope, that's the right one. So there are some reading on the throat and urine cultures, lab 7. Please read that. There's actually very little to read. 1 page on the throat cultures, and then 1 page on the oxidase test, 1 page on the catalase test. And then you need to read about the coagulase test, the coagulase production. And you can see this at the end of the lab module. This lab module has an appendix, and you should read the appendix. We have some supplemental materials in this module, video clips. The virtual lab has been removed because it requires Flash, and Flash no longer works. For this lab, the learning objectives: 1, be able to explain how to properly collect throat and urine specimens for routine culture in the lab. 2, list common bacterial pathogens isolated from the throat and urine cultures. 3, know the culture media used to cultivate these pathogens. 4, recognize which tests are used to identify throat and urine pathogens. And 5, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. Understand we will be talking about the XV factor testing, but I'm not going to hold you responsible for understanding it, nor will I test you over that material. Any question about any of that? All right. Once a specimen is received in a laboratory, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated for throat and urine specimens. Below is a table overviewing the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For throat cultures, we really only have 2 media that we use to cultivate common throat pathogens. The number 1 most common throat pathogen is Streptococcus pyogenes, and it is the organism that causes strep throat. We grow Streptococcus pyogenes on blood agar because Streptococcus pyogenes is a fastidious organism. It will not grow on a general purpose media, but blood supplies the nutrients that Streptococcus pyogenes needs. And so it will grow on blood agar. The other common bacterial pathogen we need to talk about is Haemophilus influenzae. You find this most often in young children and in infants. It is not really a pathogen of adults. It can be, but it usually is not. You cannot grow Haemophilus influenzae on blood agar. It is fastidious, but it will not grow on blood agar. And the reason it won't grow on blood agar is it does not lyse the red blood cells. And if it doesn't lyse the red blood cells, it can't get the nutrients available in red blood cells. Which, by the way, some people mention that cells use the hemoglobin and the iron released by blood. Those are only 2 of the nutrients released by blood. Haemophilus influenzae does not need hemoglobin or iron to grow. It needs NAD, which is, I think, the factor V. And what's the other factor? Maybe the factor 10. It needs 2 other factors that are supplied in the blood cells, but Haemophilus influenzae cannot use them because it does not lyse the red blood cells. And so those factors are not released into the media. So Haemophilus influenzae will not grow on blood agar. It is fastidious, but it will not grow on blood agar. You can get Haemophilus influenzae to grow on chocolate agar. Chocolate agar is the same as blood agar. The difference is blood agar adds the blood when the blood is allowed to cool to 50 to 55 degrees. And that's why the blood is whole in blood agar and you can see blood cells. Chocolate agar, we add the blood cells to the agar immediately when the agar is pulled out of the autoclave. So the agar will be around 100 degrees Celsius. And when we add the blood at that temperature, that lyses the blood and it changes the color from red to brown. And that's why it's called chocolate agar, because it looks like chocolate, but there's no chocolate in it. It's actually blood that's in it. Anyways, that lyses the blood cells and that releases the factors that are required by Haemophilus influenzae to grow. Any question about any of that? All right. For urine cultures, we grow them always on two media, even if you only have, like one urine sample from one patient, you always grow that on two media. We first grow it on blood agar, and that will obviously grow fastidious organisms and gram positives and gram negatives. We also grow from that same patient the culture on either McConkey's agar or EMB agar. McConkey's agar and EMB agar only grow gram negatives, but the most common pathogens found in urine cultures are gram negatives. The family Enterobacteriaceae is the main family causing the pathogens, and we can also call them coliforms because they're coliforms that grow in the colon. The number one pathogen found in urine cultures is actually E. coli, which is a member of the family Enterobacteriaceae. However, there are other pathogens. There's other gram-negative pathogens in the Pseudomonas family. And there are other pathogens that are gram-positive, including various members of Streptococcus, Enterococcus, and Staphylococcus. All of those are gram-positives. And to grow the gram-positives, we have to get them from the blood agar. The gram-negatives we can get from the blood agar, but it's easier getting them from the McConkey's agar or the EMB agar because these plates will only grow the gram-negatives. Any question about the common bacterial pathogens found in the throat or a urine culture? All right, let's talk a little bit further about throat cultures. Sore throats are caused by viruses and bacteria. If the sore throat has white spots on the sore throat, especially in the back or in the tonsils, white or gray spots on the tongue are often seen. And then the throat will be red. That is a bacterial infection. On the other hand, if it's a viral infection, the throat will be swollen and there will be redness, but there will not be those whitish spots. So a clinician often looks at the throat to determine if it's a bacterial infection, in which case they may give antibiotics. But if it's only a redness and swollen in the throat, it's likely a virus, and the clinician will not prescribe antibiotics because the antibiotics will not help against a virus. The most common bacterial pathogen found in sore throats is Streptococcus pyogenes, the organism that causes strep throat, and it is also named Group A Streptococcus. I don't use that name. That's using the antigens of Streptococcus, which that's an old system. I use the scientific name, and it's also abbreviated GAS for Group A Streptococcus. And I certainly don't use that name. Strep throat can be diagnosed by swabbing the back of the throat and the tonsils. Those are the good regions to swab. You do not want to swab the sides of the mouth, the tongue, or the top of the mouth. A tongue suppressor helps by keeping the tongue away from the swab. Patients who test positive for Streptococcus pyogenes are almost always placed on antibiotic therapy. The reason being is most patients cannot throw off strep throat. Streptococcus pyogenes has a capsule around the cell, and that capsule tends to protect the bacteria from the immune system. Not only can the capsule hinder antibodies from binding, that's part of the immune system, binding to the bacteria, but it also prevents the white blood cells from phagocytizing the bacteria. So a capsule will prevent the white blood cells from being eaten by white blood cells like monocytes and macrophages. If you've ever had a strep throat, you probably know that you generally have it until you go in and see the doctor and they put you on antibiotics. Actually, once you get strep throat, you're more at risk for getting it later. And that's because the antibiotics may just knock the numbers down to very low numbers, but the immune system cannot clear the infection from the body. And so you're more likely to get it later. I got strep throat about when I was in 7th grade, and I got it about 5 times later up until I was in college. Fortunately, after that, I never got it again. Streptococcus pyogenes is fastidious, so it needs to grow on blood agar. And what we do is we swab the blood agar plate from the throat culture, and then we allow that plate to grow, and you look for small white colonies that have clear, complete zones of hemolysis around them. That's beta hemolytic activity by the colonies. And that's likely to be Streptococcus pyogenes, but it could be another, excuse me, I went too far, another Streptococcus or some other beta hemolytic organism. Usually you can tell Staphylococcus aureus from Streptococcus pyogenes because the colony of Streptococcus pyogenes is very tiny and it's a small white colony. Staphylococcus aureus colonies on blood agar are fairly large and yellowish in color. And then there are some other Streptococcus species that do cause complete beta hemolysis, and it's very difficult to distinguish them from Streptococcus pyogenes. So we need to do some tests to prove that this is Streptococcus pyogenes. So first, here is a throat swab that is showing you alpha hemolysis, and the alpha hemolysis is very weak. You only see it, the darkening, where the bacteria are very heavy, and in this region here, it's not very obvious. If I blow it up really, really large, I can see a little bit of alpha hemolysis around this colony, but around most of the other colonies, it's very weak or absent. Shrink that down a little bit. Anyways, this is not beta hemolysis, so it's not Streptococcus pyogenes. And where's my scroll bar? Here is Streptococcus pyogenes. You see the little white colony right in there, right there? That's the colony, and it's surrounded by a fairly large clear area, which is complete hemolysis of the blood. And you can see complete hemolysis of the blood all over there, even there and there and there. Once we have complete hemolysis of the blood, meaning beta hemolysis, and it looks like a small colony that could be Streptococcus pyogenes, there's only 2 tests we need to run. to show that the bacteria is Streptococcus pyogenes that's causing the sore throat and the beta hemolysis. First, we run the catalase test. Catalase is an enzyme that converts hydrogen peroxide into water and oxygen. The production of the oxygen causes bubbling in the hydrogen peroxide. So we look for bubbling. And so this will be bubbles in the hydrogen peroxide. This is a catalase positive test. This is a catalase negative test. There are only two families that grow in the air in a petri dish, meaning a normal petri dish with just the lid on top, that are exposed to the air, that are catalase negative. That's the family of streptococci and the family of enterococci. So they're catalase negative. Once we have that test, we then further do the basin susceptibility test, where we take the beta hemolytic bacteria and streak it over this area of the plate or all of the plate. And then 5 minutes later, you put on the disc containing the antibiotic bacitracin. And then you incubate the plate and you look for an area with a zone of inhibition of a certain size. And if there is a zone of inhibition of a certain size, this is from Streptococcus pyogenes. It is sensitive to bacitracin. The other members of the family Streptococcus are not sensitive to bacitracin, and so they will be bacitracin negative in their test. Any question about that? All right, another organism that is clinically significant when isolated from the throat is Haemophilus influenzae, a gram-negative coccobacillus. Haemophilus influenzae is mostly seen in small children and infants. It is one of the most common causes of ear, eye, and sinus infections in babies and young children. Haemophilus influenzae is fastidious, but it will not grow on blood agar because it cannot release, lyse the blood and then release the factors in the blood. Two nutrients that are required by Haemophilus influenzae, which are in blood, but are not supplied by an unlysed blood cell, are NAD, which is the V factor, and hemin, the X factor. However, Haemophilus influenzae can grow on chocolate agar because the blood cells are lysed, and so those nutrients are released into the agar. You can get Haemophilus influenzae to grow on blood agar if you streak Staphylococcus aureus down the blood agar, which is this strike there. I'm not sure why that's a white strike because Staphylococcus aureus is normally yellow, but that streak there is from Staphylococcus aureus. And then you streak the Haemophilus influenzae parallel to it in this region here. And you'll note that in this region of the plate, there is no growth of the Haemophilus influenzae. The only growth of Haemophilus influenzae is near the Staphylococcus aureus. And the reason why the Haemophilus influenzae is growing in this region is because the Staphylococcus aureus is lysing the red blood cells, which is releasing the growth factors required by Haemophilus influenzae. And then the growth factors are diffusing out to about here on the blood agar. Any question about any of that? Let me shrink this down. All right, let's move on to the urine cultures. Urine cultures are used to diagnose urinary tract infections. These include Cystitis, that's a bladder infection. Urethritis, urethra infection. Polynephritis, which is a ureter infection or a kidney infection. If you do not know, the bacteria can move up from the urethra to infect the bladder, and can move up the bladder to infect the ureters, and can move up the ureters to infect the kidney. And a kidney infection is very serious. Any question about that? The urethra infection is not serious, but it is irritating, and so you'd want it removed. But the kidney infection is really severe. We'll just word it that way. Females are more predisposed to developing a UTI, a urinary tract infection, than males, simply because they have a shorter urethra. The genital area, to get a urine sample, the genital area is clean prior to collection. The patient then begins voiding, but does not collect the urine initially. You wait until the urine is midway through its flow, and then you collect the urine sample in a sterile container. This reduces the cultivation of organisms from the urethra. The urethra is loaded with microbes, and if you simply were to urinate out of it, the urine will contain bacteria from the urethra. Furthermore, if you remember, I did mention that in females, they have normal microbiota, bacteria and other microbes that are actually growing in their bladder. I've looked to see if males have a normal microbiota in their bladder, and I can't find anybody who's done any research or looked. And so I don't know if males have normal bacteria in their bladder or not. And it's funny because people have known about women now having bacteria in their bladder for at least 10 years. And like I said, everyone's done the study on women. So I don't know if males do or do not. It's not terribly important, but my point is that even if you were to sample urine directly from the bladder, there will be bacteria in it, assuming that the urine comes from a female. And for males, I just don't know the answer. Okay, but there will be a lot of bacteria coming from the urethra. Even if you clean the genital area prior to doing the sample, and you do the clean catch midstream, catching the urine midway through. You can submit a catheter up the urethra into the bladder, and that will be cleaner. The trouble is the catheter can pick up bacteria from the urethra and then transfer the bacteria to the urine in the bladder, and then you'll get the urethra bacteria that way. So even the catheter is not, what's the word, sterile. You will get bacterial contamination using a catheter. You could directly sample the urine by a suprapubic aspiration, putting a needle through the abdominal wall into the bladder and directly sampling the urine. That will avoid any contamination from the urethra, but in females, we can get bacteria from the urine in the bladder. Okay, at least from females. We might from males, I just don't know. Anyways, these are the three ways to collect a urine sample. The suprapubic aspiration is the cleanest way, but patients are not going to like it. They're going to hate getting a needle, and they don't really like catheters either. And they don't mind the clean catch midstream method. So that's why we frequently use that method. for getting a urine sample. Once you get a urine sample in the lab, let me make sure I've discussed everything. Yeah, I have. We then, once we get a urine sample, put 0.001 milliliters of urine onto the blood agar plate using a special inoculating loop that actually measures 0.001. Did I mention that enough? 0.001 milliliters. That's 1 microliter of urine. And you put that, streak it here, and then streak it like that. So you'll get something looking like that on the blood agar plate. You then also take the urine sample and put it on either a McConkey's plate or an EMB plate to get the growth of gram negatives. The interpretation of a positive urine culture is multifaceted. Some factors that need to be considered include the type of collection. You'll get more contamination from the the clean mid-catch method than you will from a catheter, than you will from the suprapubic aspiration. The gender of the patient will also determine the number of bacteria seen. Generally, females will shed more bacteria from their urethra than males. And then the number of colonies per mil of urine is considered, and the presence of polymicrobial growth. If there's multiple organisms isolated, it may indicate a poor collection technique, like the patient may have done a real poor job of sterilizing their genitals before collecting the urine. Any question about any of that? All right. Once you get the bacteria growing on the blood agar and or the McConkey's agar, if it's gram negative, you'll see it on the McConkey's or the EMB plate, and both gram positive and gram negative will grow on the blood agar. You then run some tests to try and identify the species of bacteria which is causing the patient to be sick with the urinary tract infection. We can test for the coagulase test. This is looking for the enzyme coagulase. This enzyme is used to differentiate Staphylococcus aureus from other members of the staphylococcal family. Staphylococcus aureus is coagulase positive. Other members of the Staphylococcus family are coagulase negative. What this test does is the enzyme causes the plasma or the blood to clot. Exactly, the coagulase takes the fibrinogen in the plasma and creates fibrin, which are the clotting proteins, and then they will cause a clot to be forming. Staphylococcus aureus has the enzyme coagulase to help protect the bacteria from the host immune system when the bacteria is first causing an infection. When the bacteria are low in number, they're most likely to be removed by the immune system. So Staphylococcus aureus can cause a clot, and then the clot will protect the bacteria inside the clot. The host immune system will have to phagocytize the clot, eat away the clot, before the white blood cell can get to the Staphylococcus aureus. My point is, this enzyme is a factor in the pathogenicity of Staphylococcus aureus. And that's why Staphylococcus aureus is one of the reasons why, I should mention, one of the reasons why Staphylococcus aureus is more pathogenic than other members of the staphylococcal family. Staphylococcus aureus can have other pathogenic factors besides coagulase, but that's one. You can determine the coagulase test in a test tube, the tube coagulase test, by if the bacteria produce the coagulase, will get the plasma to clot, and then you put the tube and put it on its side, the clot can be seen there. And if the plasma with the bacteria in it, and the bacteria coagulates negative, the plasma will not clot and it will remain a liquid, which is shown here by the coagulase negative tube. For the slide test, meaning we have the tube test and the slide test, you can put the plasma and the bacteria together, and if the bacteria coagulates positive, they'll cause the plasma to form clots, which will form the dots and the clumping shown here. And if the bacteria are coagulase negative, the plasma will remain a liquid and will be this milky, smooth color. Any question about that? All right, so that's one test we use for determining what the pathogen is. There are other tests we use to determine the pathogen, and the most common culture, the most common pathogen in a urine culture is actually E. coli. E. coli is a member of the family Enterobacteriaceae. It's a gram-negative species. There are two families which are gram-negatives, which cause a urinary tract infection. The family Enterobacteriaceae, and then the family Pseudomonaceae. We can distinguish between these two families of gram-negatives by looking at the oxidase test. The family Pseudomonaceae, including the pathogen Pseudomonas aeruginosa, is oxidase negative. The family Enterobacteriaceae, including E. coli, are oxidase negative. This is the most common test we use to separate these two families. But we could use other tests, meaning the Pseudomonaceae are gram-negative bacillus that are aerobic. The Enterobacteriaceae are gram-negative bacillus that are facultative anaerobes, meaning they will prefer to grow in oxygen if oxygen is present, but if oxygen is not present, the cells will continue to grow by anaerobic respiration. They also differ in their glucose fermenting capabilities. The family Enterobacteriaceae, all members of the family, can ferment the sugar glucose. The Pseudomonaceae family, all members of the family, cannot ferment the sugar glucose. So we call that a non-glucose fermenting species. Or in this case, the family is totally non-glucose fermenting. All right, any question about the oxidase test and how you can distinguish the Pseudomonaceae family from the Enterobacteriaceae family? Let's talk a little bit more about the oxidase test. The oxidase test is looking for the presence of cytochrome C, which is in the electron transport chain of an organism performing aerobic respiration. So if the organism can perform aerobic respiration, it'll have cytochrome oxidase C, which gives rise to the oxidase test. And Pseudomonaceae has oxidase, and it will show up purple or a blue color in the oxidase test. E. coli, which is oxidase negative, will not show up in the oxidase test, and it will not show up blue or purple. Any question about any of that? All right, so the oxidase test is the main test to distinguish between the family Pseudomonaceae and Enterobacteriaceae, which are both small gram-negative rods. All right, there's one more test I think I need to talk about. And it's not here, it's the catalase test. Oh, I've already talked about the catalase test. I talked about the catalase test with the throat cultures. And that is, you look for the presence of the enzyme catalase. If you have the enzyme catalase around, you see bubbling. If catalase is not around, you will not see bubbling. All right, let's move on to the summary. This lab is discussing the main pathogens of urine and throat specimens. In this lab, there's going to be videos. You're not going to be performing any of these activities. However, you will be held responsible for knowing how to perform them. Here we have a video on the catalase test, a short video, and a short video on the coagulase test, and I believe an even shorter video on the oxidase test. We used to have a virtual lab exercise to do. However, that lab uses Flash, which no longer runs, so we've removed it. And then let's go to the worksheet. In this lab, you only have 13 or 14 questions, although 13 has like 5 questions to it. So this lab is a fairly easier lab. It should take you less time. It has no case studies. I believe all labs from this point, meaning all future labs, will have some case studies, and they're a little harder, and they take some time to solve. So for question 1A, it's referring to the presence of the enzymes catalase, coagulase, and oxidase. Which of the enzyme converts fibrinogen into fibrin? For 1B, name an organism that tests positive for this enzyme. For 2A, name an enzyme that detects the presence of cytochrome C proteins in the electron transport system. And for 2B, name 2 organisms that test positive for this enzyme. These organisms are mentioned in the lab module as well as in the video clip. For 3A, name the enzyme that hydrolyzes hydrogen peroxide into the non-toxic products water and oxygen. And 3B, name 2 organisms that test negative for this enzyme. You can either mention 2 species, or in this case, you can mention 2 families, because the entire family is negative. 4, also referring to the enzymes catalase, coagulase, and oxidase, which of the rapid tests, meaning which of those 3 enzymes, is used to differentiate between the following: A, Staphylococcus and Streptococcus; 4B, Staphylococcus aureus and other members of the staphylococcal family; and then 4C, which enzyme is used to distinguish between Enterobacteriaceae, such as E. coli, and the family Pseudomonaceae. 5, concerning specimen collection for routine throat culture: A, what areas of the throat should be swabbed, and B, what areas of the throat should be avoided, the mouth. For a 6, what is the most common cause of bacterial pharyngitis? Pharyngitis is a sore throat. We're referring to a bacterial sore throat, so do not tell me the human cold, okay? Because that's not a bacteria. 7, What test is used by microbiologists to differentiate between beta hemolytic group A streptococcus and other beta hemolytic streptococcal species isolated in throat cultures? You do not need to run the catalase test here because we're only looking at members of Streptococcus family, and all members of the family are catalase negative. Any question on that? All right. 8, answer the following concerning a child's throat culture. A, state 2 non-selective agar plates that are inoculated. 1, 2. And 8B, what organisms can be isolated on CAP, but not on BAP? CAP standing for chocolate agar plate and BAP for blood agar plate. 8C, you do not need to answer because it's not discussed in the lab module. If you care to answer it, you'll have to do your own research online. You will not get extra credit if you answer it, but you may if you want. 9, list 3 methods that are used to collect urine for routine culture. 10, list 3 reasons why urine specimens should be rejected for culture. And that is discussed in the lab module. You can also do a search online. 11, how much urine is placed on the surface of a blood agar plate? 12, what is the most common bacteria isolated from patients with cystitis? Remember, that's a bladder infection. We're talking about a species of bacteria. 13, identify the Gram reaction, that's the Gram stain, and the cell morphology, that is the shape of the bacteria, not the growth arrangement. So 13A, list the Gram stain of Staphylococcus saprophyticus and the shape. And that should be mentioned in the lab module. For 13B, do that for E. coli. For 13C, do that for Pseudomonas aeruginosa. For 13D, do that for Streptococcus pyogenes. And then for 13E, you can do this one, Haemophilus influenzae, from both the lab module and the appendix of the lab module. Let me show you the appendix of the lab module just briefly. In the appendix, we got a little bit talking about the family Streptococcus. And then talking about the beta hemolytic streptococci, which are mainly Streptococcus pyogenes, but there are other members that are beta hemolytic. And this is discussing them, mainly Streptococcus pyogenes, which does cause strep throat. Nowadays, scarlet fever, rheumatic fever, and even impetigo are rare. I think how do we say that? Erysipelas is also rare, but Streptococcus pyogenes causes all of these. Scarlet fever and rheumatic fever only happen after a patient has had strep throat, and then the bacteria invade the beyond the throat and start invade the blood and then cause the other symptoms. So these are actually rare nowadays because we do treat Streptococcus pyogenes with antibiotics. But before antibiotics, scarlet fever and rheumatic fever were very common infections, even in the first world country such as the United States. All right, we have a little discussion of Haemophilus influenzae. And then lastly, we discussed a little bit about the coagulase test. All right, any questions on this lab? If not, let me remind you that I'm here to help you in the lab. if you're struggling and getting a lot of answers wrong in the first grading of the lab, you should probably use the help I'm offering. Just if you have a question when you're answering the lab, just ask. And I will, at the very least, point you in the right direction. Sometimes I'll give you the direct answer if it's a complicated one. If it's one that I feel you should know, I'll just give you hints and point you in the right direction. But I will help you. Okay, that's why I stay here in the lab. So use me if you need to. And I will be here until 8:30 or the last student logs out. So far, only once have I ever logged out on a student because the class was long over. And I told the student, do you have any questions? Because the class is now over and I want to end it. All right, any questions? If not, go ahead and start working on the lab. I'm here until 8:30 to answer